also have an interest in non-invasive brain stimulation. So first of all, I would like to thank you for the opportunity of being here today. And I will introduce the speaker and manage the question session later on. Just a few words, I won't take too much time. Twin NIBS, this symposium today is a Twin NIBS symposium. Twin NIBS is a European project that aims at advanced non-invasive brain stimulation. And to do so, it aims at fostering collaboration between Serbia, Italy, Germany, Austria, and Denmark. How they do that? They do through short-term visit, workshops, and boot camps, and therefore this symposium fits perfectly within the scope of the project. The title of the project, of the symposium, it's Non-Invasive Brain Stimulation and Beyond. Today we will address kind of two topics. The first one is on the challenges of non-invasive brain stimulation, and the second part will focus more on in broader sense, on the challenges of women in neuroscience and the steps that have been taken within the Twin NIBS project to address these challenges. Just a couple of points. The symposium is in blended modality, so we will have some speakers online and some in person. And concerning questions, I'd suggest that we start with the talks, and then at the end of each talks, if we still have time within the slot, we can ask a couple of questions. Otherwise, we will keep them for the end. Our first speaker is Armita Faghani. She's a postdoc fellow at the Danish Research Centre for Magnetic Resonance. Her talk title is EEG-based monitoring of intermittent theta burst stimulation, effect targeting the left dorsolateral prefrontal cortex. So I will leave now the stage to Armita.
Yes, thank you. Can you just see my screen?
Yes.
Okay, perfect. So, hi everyone. First, I want to thank you, the organizing committee, for the interesting symposium and also inviting me and giving me time to just share the latest contribution to NIBS field. And I'm Armita. I'm postdoc fellow at the RCMR, Danish Resonance Centre for Magnetic Resonance. And today my talk will be about the EEG-based monitoring of ITBS, targeting the left dorsolateral prefrontal cortex, which this is part of bigger project, Precision Brain Circuit Therapy, funded by Innovation Fund in Denmark and led by Professor Hartwig Siebner. If I can just go. Yes, if I just want to start my talk with brief introduction to TMS and its application. Most of us may know that TMS or transcranial magnetic stimulation is a non-invasive brain stimulation method that allows us to modulate the neural activity and a functional brain network at the time of the stimulation and beyond the time of the stimulation. And the way that it works, it depends on the place that you put the coil. The varying magnetic field is used to induce the electrocurrent below the coil. And depending on where you are putting the coil, if you put it close to the motor cortex, you have a well-known readout MEP, a motor evoked potential, if you have the enough strong TMS pulses. And if you just move a little bit your coil to more, for example, frontal area, you will have another readout, which call it TEP or TMS-evoked potential, which is direct response of the brain to the stimulation. TMS can also be used as a therapeutic intervention for neurological and neuropsychiatric disease, for example, like chronic pain and specifically depression. If you repeat the stimulation, instead of single parts, you repeat the stimulation, we call it repetitive TMS or RTMS. and we apply it to the left dorsolateral prefrontal cortex. It has been approved by FDA that it's been helpful for patient, specifically patient with treatment-resistant depression. But it depends on the pattern that you are choosing to deliver. These patterns have been tried during the time. It's been low frequency, RTMS high frequency. You can deliver more than a single pulse. You can deliver a train of pulses. Each time we have theta burst stimulation in a continuous and intermittent pattern. And beside the stimulation pattern, the target is also plays a major role. And here you can see the evolution of the TMS targeting during the time that has been started with the scalp measurement to the recent coordinate that has been introduced by measuring and calculating the functional connectivity. Considering all of these factors, recently Nolan Williams and his colleague has introduced new protocol, accelerated protocol called Stanford Accelerated Intelligence Neuromodulation Therapy, or SAINT, which they have delivered 10 ITBS sessions per day for 5 consecutive days, and they relieved promising results in suppressing the depression symptoms. Considering all of these factors, as I mentioned today, my talk would be about the bigger project, Precision BCT, which is Precision Brain Circuit Therapy, that we aim to develop a personalized brain stimulation approach for a patient with TRD, treatment-resistant depression. And we are trying to leverage the methodological approaches and advances to further optimize this SAINT protocol. What we are doing is to use functional and structural brain MRI to individualize the site and also the intensity of the stimulation. We use functional connectivity and also online E-field calculation to have the intensity and the location of the stimulation. And more importantly, we use functional and structural brain mapping methods to understand how this stimulation works and what is the relationship between these changes that can happen on the brain network and the clinical improvement. And we use EEG here, the EEG-based monitoring for the safety concern and also for the target engagement. For the safety concern, TMS has been considered as a safe therapeutic modalities, and it has few adverse events, and this is mainly about the headache most of the time and discomfort at the site of the stimulation. But rarely TMS-induced syncope or seizure has also been reported. It's really rare, below 1%, but because of the dramatic growth in use of this intervention in both existing and also novel pattern, which are more complex, with higher frequency, with higher intensity, the importance of utilizing a safety software and monitoring the subject during the experiment, they're gaining more attention. Because of that, we have introduced STIMO, EEG-based stimulation monitoring, which is developed by my colleague, Adam Ibistruk, and I also help him to modify it, to do some modification, and also do quite several experiments to validate the software. And this is some graphical GUI, graphical user interface, or GUI Python-based and open source software that calculate the features from the EEG signals, the continuous EEG signals online. And as you can see here, this is the main window of the software after running this. And you can choose the channels that you want to calculate the features, and also the channels that you want to stream and you are interested to monitor during the experiment. The features that you can choose and is already available in the software, there are based on literature about the automatic online seizure detection for a patient with epilepsy, And this software has been developed for a TMS protocol, but it's possible to be used with other stimulation methods such as ultrasound and so on. Here also can show you that for using the software, you need the calibration and also you need the normal values or threshold for each features that you have been selected in first step. These values can be extracted when the subjects are in the same state as their intervention. For example, the resting state. So you go for calibration mode, and after you can see here that there would be 6 plots of distribution for 6 selected features, and they will be updated every second. And after you are done with the calibration, you can see that the safety and threshold are established. Here there are the red lines for each features. And after starting the intervention phase, after a specific defined time of delivering the triggers, every second each features would be calculated, and if there are between the threshold, you will see something like this on your screen, or if something happened and the features exceed the threshold, the background would be in red. And for the target engagement, we integrated a setup for a real-time recording of brain activity with EEG during the RTMS session. And for that, first we acquired T1 and T2 images, and then we continuously recorded 3 minutes resting state 3 times before and 5 times after of ITBS intervention. The ITBS lasted 10 minutes. It was 2 seconds on and 8 seconds off of the stimulation. And it has been delivered in DLPFC with neuronavigation system. And we had 25 subjects in active group, which has been divided in 2. Half of them has been in the group that they received the beep sound before receiving each time receiving the train of pulses. So they were aware about receiving a stimulation. And half of them, that we call them no beep group, there was no auditory cue. For this case, we want to study that is there for the auditory cue has any effect on the brain state that can change the effect of ITBS or no. And we also have covered the sham group. The whole aim was to study the effect of ITBS within the consecutive trains for the immediate effect, and also after the whole session to just study the after effect on the cortical oscillatory activity. I will also share a little bit of the preliminary results here. We calculate the power spectrum of 3 different frequency bands for all electrodes and for each time point. And then we do the comparison by using cluster-based permutation test. And as the 2 groups, the beep and no beep, there were no significant differences between them. We pulled the data together, and here you can see the result of 25 subjects altogether. Here I'm showing you the changes in the power for each time post time compared to the last pre. So it's post 1 minus pre, the last pre, the post 2 minus last pre, and so on. And the statistical test has shown that sorry, that there's only significant suppression in the theta power right after the stimulation post 1, and it's mainly in frontal channels. And for the rest, there was not significantly differences. And then we try to see that which channel has experienced the most significant alternation, and it was the channel below the coil. And here you can see the relative power of all resting state measurement for the theta band and also for the channel below the coil. And here you can see that the same effect that we have seen in the previous slide, the suppression, the significant suppression happened, and it also shows the relative power for all subjects. And we also check that how was this alternation in 2 groups, the beep and no beep. Here also you can see that the effect right after stimulation is the same, the suppression effect. And we can also see here the inter-individual and intra-individual variation over the relative power in this band between these two groups. In summary, we have so far developed a real-time EEG-based brain state monitoring software, STIMO, for a safety aspect of brain stimulation session. And the preliminary result of ITBS of the left DLPFC has shown a short-lasting but widespread over the frontal, mainly in the frontal channels of suppressive effect for the resting state oscillatory activity, and it was mainly only in the theta band. But for sure, further validation of ITBS-induced effect that what we have seen in comparison with sham group should be evaluated and is also under investigation. So I want to thank my colleague at DRCMR, an innovation fund in Denmark, for supporting this project, and NIBS and Toronto Symposium for giving me a chance to share my work. And I would be glad to discuss if there is any question.
Thank you, Armita.
Come.
I think we have a couple of minutes if there is a question now, otherwise we'll keep them for the end of the symposium. No? I guess we will summarize everything at the end then.
Sure.
So we can move on to the next speaker. Our next speaker is in person and is Dunja Paunovic, yes. Right? Dunja is a PhD candidate at the University of Belgrade, and she's studying the neural underpinnings of human memory processes by combining non-invasive brain stimulation, EEG, and behavioral measures. Oh.
This guy, yeah. Yeah.
Yeah. Would you like to start with the...
No, sorry.
The listeners? No.
We shared the program and not the slide.
Oh, okay.
This one.
Do you prefer to stand up? We can move the microphone. It's up to you. If you prefer to stay, yeah. It seems more natural. Is this now fine? Should I move this?
Can you add one more?
And then five formatting close, and Armita I can put here or
yes,
this.
Yeah.
Okay. So can I start? Yes.
Yeah.
So hi, I'm Dunja, and today I'll present you the study from my PhD project that is supervised by Jovana Bjekic, which is she is also a PI of the Twinings project. So I am very grateful to be a part of the project and invited here to present my work. In this study, we aimed to modulate associative memory processes by implementing TES and EEG methodology. Let's first start with introducing core concepts around which we build the whole project. First one is, of course, our target construct, associative memory, which we define as the ability to bind unrelated information into meaningful units and encode them as distinct memories. To put it more in an everyday life setting, basically when you're trying to memorize somebody's name and to put it in like the whole episode of that person, so associated with their face, for example, you would rely on associative memory. The second important or core concept is theta hypothesis. What is theta hypothesis? Basically, that's the assumption that brain activity that we detect in EEG signal that ranges between 4 and 8 hertz could be a neural underpin of mnemonic function.
because it co-occurs with a situation in which we probe memory. For example, in human subjects, when we give them memory tasks, and even in animals, when they are doing like maze tasks and trying to orient and learn the space. And the third important core concept is frequency modulated TES, which we know as transcranial alternating current stimulation, and the new one that we are introducing in this study, which is oscillatory TDCS. That's how we called it, but basically it's very similar to TACS. It only oscillates in a positive polarity of the electrical field. What was our idea? But basically, if we mimic the natural frequency of function, in this case associative memory, and natural frequency being in theta's range, that we would maybe engage functionally relevant circuits in more effective way. So let's continue and dive into associative memory a little more. So when we assess it, we are relying on two essential memory subprocesses, encoding and retrieval. Encoding we define as a process of memory formation, and retrieval as a process of accessing already memorized content. In our studies, we rely on paired associate paradigm, and we probe encoding process by exposing subjects to pairs of previously unrelated stimuli. In this case, these are the photos of faces and concrete nouns. We instruct participants to memorize as much as they can. Then we probe their retrieval processes by asking them to try to recollect the word by probing it partially with just a photo of a face from a pair. And after that, we also test their ability to recognize by asking them to classify previously seen pairs from those who are wrongly paired. So from this paradigm, we extract two markers of associative memory performance: recollection ability and recognition ability. When we try to see how these markers are reflected on neurophysiological activity, we can find in the literature two important paradigms. One wanting to tackle underlying activity during the encoding processes, and the other one trying to tackle activity during the retrieval process. The first one related to encoding is called subsequent memory effect paradigm. Basically, studies incorporating this paradigm found that activity in the theta spectrum is more powerful for the trials that are subsequently memorized when contrasting to the trials that are subsequently forgotten. But activity is higher during the learning. So that's how we tackle the encoding. And the similar logic is applied when exploring the retrieval processes, but that paradigm is called old new memory effect paradigm. So why did I tell you all of this? Because we used it as an input when developing our oscillatory protocols. Basically, we had an idea to mimic the rhythm of our stimulation with natural theta rhythm. And here on the slide, you can see two examples. One is a tax example, and one is just oscillating in the positive polarity. We expected that maybe if we compare these protocols with the standard constant TDC protocol, also a sham, that we could see more pronounced effects on either behavioral or neurophysiological level. I'm foreshadowing a little bit. Also, I wanted to guide your attention towards one more thing, because I was referring to theta activity as a range, that it varies between participants. And we know that in whole NIBS field now. We are moving towards that one size does not fit all approach. So in order to be sure that we are mimicking the person's actual dominant frequency that we associate with memory, we used a subsequent memory effect paradigm to extract the dominant frequency for each person, and then we designed this oscillatory protocol for each participant individually. Yeah. Okay. So let's just recap. So here you can see the study goal and the important questions that we wanted to answer with the study. So we wanted to see, can we detect previously detected changes on behavioral level on associative memory, of course. Also, can we detect changes when we take into account neurophysiological markers of associative memory? Hopefully see, do the effects depend or differ in different TES conditions, so different oscillatory protocols, and also compared to active TDCS control protocol. And what also this design gives us insight in is, do we possibly modulate encoding or retrieval processes when we are detecting effects on, for example, behavioral level? So yeah, this slide is just to avoid confusion because there's a lot of EEG in this study. So we use EEG measurements in 2 stages. The first is phase 0 that I already mentioned, that we use task EEG to extract the individual frequency. Then we apply different TES protocols, but then we also use task EEG to detect, to observe and possibly detect effects on behavioral as well as neurophysiological level. This is more detailed description of the whole study procedure. So first, we recruited 42 subjects. Then it was that phase 0 in which we extracted individual theta frequency. And you can scan the QR code if you want to see the whole procedure more in detail. It's a publication. After which each participant underwent the experiment. So they underwent each TES condition in a contrabalance order on separate days. So sessions were had a break in between. And after each condition, we assessed their memory performance on paired associate paradigms. One was that we standardly use, so it was a face word paradigm. And the other one was more exploratory that we wanted to first test in this study. So it's a paired associate paradigm, but with objects like visual stimuli instead of words. We wanted to see how it will be compared to the verbal stimulation. And I wanted to mention that our locus of stimulation is from parietal regions with expectation that we would kickstart the activity in frontal parietal network that relays through hippocampus, and hippocampus is the core structure that we associate with memory. Here are the first behavioral results. So you can see in first big column, it's a face word results from face word paradigm. And the second big column is results from face object paradigm. In the first row, you can see the recognition measure outcomes. Second row, cued recall. And the third row is repeated cued recall. They learned once more, and then we tested them once again. You can see like the first small column is sham, then it's TDCS, then theta and tax, and that repeats in every plot. So here, group level comparisons of each condition against sham did not show any difference in performance. So we basically did not register effects on behavioral level. We can also see that we detected variability across tasks as well as task measures, and that is something that we wanted to try to tackle, but diving deeper into EEG analysis later. So not only that we are left to see whether we detected some changes on neurophysiological level, but also maybe to use these results to uncover the criteria for detection different patterns in responsiveness to TES. So I will leave you here because this is all the part that we are currently on in this study. And also I want to thank everyone once again and our collaborators from faculty from philosophy that took part in this project. Also our whole group of human neuroscience from IMR and Science Fund that funded this study. So, yeah, that's all.
I may have one quick question.
Oh,
and perhaps it's what we're planning to do already, but I was wondering, so you mentioned that the feature that you focus on is the theta power, and the higher the theta power, the better performance. And what you're trying to do is to modulate this amount of theta power. So I was wondering if you're planning to look at the individual theta power before the intervention and then see whether individual who already has a higher theta power than other are, I don't know, maybe less likely to improve because they're kind of reaching a kind of ceiling.
we have their data from the session 0. So we have their individual power in theta spectrum without an intervention. So we could also calculate that measures, and in contrast, in addition, we also recorded resting state prior and after the interventions, so we can actually monitor the whole spectrum, how they changed because of intervention and what are the inherently values of theta. So we could control for that as well and see how our sample behaves. But that would be like more for purposes of exploratory analysis and possible interpretation later. We thought about that as well.
The question is whether we can, how much you can improve or you can increase.
That's an interesting way of putting it. Thank you.
Any other question? No? Okay, then we move to the next speaker. Thank you. Next speaker is Arianna Brancaccio. Arianna is a former PhD student at CIMEC. She just defended her PhD a couple of months ago. She's interested in stroke and anti-aging, and she focuses on brain state-dependent approaches in the analysis of TMS-EEG data to address the heterogeneity of findings in the literature. The title of her talk is Brain State-Dependent Approaches, How to Reduce Variability in Non-Invasive Brain Stimulation Experiment and Increase Control on Non-Invasive Brain Stimulation Effects.
Okay, thanks. Thank you, Sara. Okay, so thank you very much. My name is Arianna, and today I'm going to talk to you about brain state-dependent approaches to the analysis and recording of TMS-EEG data. And brain state-dependent approaches are exactly an approach that takes into account the state of the brain at the moment of stimulation in order to best predict for the effects of stimulation. And in this way, it is thought to be able, let's say, to reduce variability in the results of stimulation studies. And in fact, we have a problem with variability in the results of stimulation studies of both inter and intra-individual variability. Many works have tried to address this issue. For example, this is a paper published in 2017 in which the authors collected evidence from many different papers using large cohorts of participants and comparing different stimulation protocols. And what they concluded is exactly that studies using larger cohorts of healthy subjects confirm that there is a considerable inter and intra-individual variability in response to all stimulation protocols. And this is not the only paper that came to that conclusion. Actually, there are many other papers that have concluded the same exact thing. So we have a problem with variability. And one of the potential sources at the basis of this variability has been found exactly in the state of the brain at the moment of stimulation. And this is an approach that was proposed in 2018 by Zrenner and colleagues, in this paper, they exactly say experiments suffer from high inter-individual variability and low effect sizes because the applied stimulation sequences are blind to the ever and rapidly changing instantaneous brain state. So how did they end up with this conclusion? So they were studying the excitability of the motor cortex, and in particular, they were investigating whether the state of the sensory motor mu alpha rhythm would modulate the excitability of the motor cortex. So how did they study that? They were doing an online experiment. So online, they were recording the signal from a C3-centered yacht, so optimized to catch the sensory motor mu alpha rhythm. Online, they would filter the signal at the individual alpha frequency of each participant, and in this way they could choose whether they would stimulate with single pulse TMS the motor cortex, either at the positive peak of the mu alpha rhythm or at the negative peak of the mu alpha rhythm of the participant. And what they ended up seeing is that if you systematically stimulate with single pulse TMS the motor cortex at the positive peak, you obtain MEPs that are smaller in size compared to the MEPs evoked systematically at the trough or the negative peak. And therefore, the trough was proposed to be a state of higher cortical excitability for the motor cortex. And actually, this approach was found to be essential also to predict the effects of plasticity in using protocols. So, for example, this is a paper published in 2019 by Bauer. They were studying the effects of low-frequency RTMS, so a protocol that we would all agree is thought to induce mainly long-term depression in the system. But they studied this approach with a brain state-dependent logic. So they were either releasing stimulation only at the positive peak, only at the negative peak, or at random place, as we would do if we were not controlling for the state of the brain at the moment of stimulation. And what they found, these are the MEPs normalized on the MEPs amplitude evoked before the application of the protocol. So what they saw is that if you release the pulses only at the positive peak, so the low frequency RTMS protocol at the positive peak or the low excitability state, you actually obtain long-term depression in the system. The same thing, even if with a low effect size happens at random place. So if you don't control for the state of the brain at the moment of stimulation. But what is the most interesting finding is that if you release low frequency RTMS at the high excitability state, so the trough, what you actually observe is long-term potentiation in the system. So this means that a protocol that was originally thought to be inducing only long-term depression can actually induce long-term potentiation if applied at the high excitability state. And also the other way around was shown. So this is a study with high frequency RTMS. So a protocol that is thought to induce long-term potentiation in the system. They studied it with a brain state dependent approach, again, online. So they were either stimulating with high frequency RTMS at a positive peak or at a negative peak. So this is the moment of intervention. And what they observed is that if you stimulate at the negative peak, you see long-term potentiation. So you see an increase in the MEP's amplitude after the application of the protocol. But that if you target the positive peak, you actually obtain a tendency toward long-term depression. So again, the same protocol changes its effects depending on the targeted brain states. And actually, this approach is useful to reduce variability, but also to understand more about cortical excitability in general. And in particular, we apply this approach in stroke patients' data. And I want to give you an example. So this is a paper published in 2020 by Chairperson. So what they showed basically is that if you stimulate the affected hemisphere motor cortex, you see in stroke patients this increased and even simplified response over the effect. These are typical TPs evoked over the affected hemisphere, which is very hyper simplified and increased compared to what you observe in general on healthy controls, which show these positive and negative deflections. So we said, can we explain better this result in the affected hemisphere of severe patients with a brain state dependent approach to the analysis of this data? So we took our sample of severely affected stroke patients, and first we want to look into whether we could find that increased TP. And this is exactly what we found. So these are the TPs evoked in our sample of severely affected stroke patients over the affected hemisphere. And as you can see, it is much hyper simplified and increased compared to what we observed in the blue line, which represents the unaffected hemisphere of the same stroke patients. And so we said, let's approach this data set, but with a brain state dependent logic. And in order to do so, we had to choose markers, metrics that would be sensitive to the cortical excitability. And first of all, we chose tEPs. Why? Because tEPs have been found to be markers of cortical excitability at the moment of stimulation. This is a paper published in 2019 by Pischedda. And what they found is that if you systematically stimulate at the negative peak on the motor cortex, and then you do the same exact thing but at the positive peak, and then you run a statistics, in this case a cluster analysis between the two conditions, you will see that the P70 and then 100 components in healthy controls will be of greater amplitude if the stimulation has happened at the trough. So the high excitability state. So this means that tEPs are sensitive to the cortical excitability at the moment of stimulation. And then we looked into post-pulse time frequency spectrograms, and in particular in alpha. Why? So for two reasons. First, because in severe patients, so the same patients who show that increased tEP over the stimulated affected cortex should be an increase in the post-pulse alpha power from the time post-stroke. So we wondered whether this effect could be somehow linked to the effect of increased tEPs. And also because the post-pulse alpha power has been found to be a marker of cortical excitability at the moment of stimulation. For example, it is modulated by TMS. And therefore, what we did, so this is our sample, we had 11 strokes. These are the severe strokes. They received one pulse TMS at 115% of their resting motor threshold of both the affected and unaffected hemisphere. We also had 10 age-matched controls. I'm not discussing the controls results now, but I just want you to know that we proved previous literature. So in age-matched controls, we found greater tEPs when stimulation happened at the trough, and also greater post-pulse alpha power when stimulation happened at the trough, so the high excitability state. So how did we do this? So we didn't do an online experiment, right? So we had to estimate the phase at the moment of stimulation post-hoc. So we have 11 strokes. First of all, the right strokes were flipped to the left side, so that now the left side is always the affected hemisphere. And then for each stroke, we computed the individual alpha frequency. And in this way, we could take each trial of each stroke, filter the signal at the individual alpha frequency. And in this way, we could train an autoregressive model to predict the phase at the moment of stimulation in that specific trial. And in this way, we ended up having 4 conditions for the strokes. So we have unaffected hemisphere stimulated at trough, unaffected hemisphere at no trough, so all the other phases except for the trough, and same thing for the affected hemisphere. And then for each of these conditions, we computed the tEPs, which were rectified, and then cluster statistics was computed. And the same thing was done for time frequency spectrograms in alpha. So basically, we're comparing the effects of stimulation in this portion of the alpha wave against the stimulation falling in these other phases. So let's go look into the unaffected hemisphere. So if the differentiation between the two excitability states is preserved within the unaffected hemisphere, we should see greater tEPs evoked at the trough. And these are the first results for the 40-55 milliseconds time window, the cluster analysis between unaffected hemisphere stimulated trough versus no trough. This is a positive cluster, so this means that the activity was actually greater when stimulation happened at the trough, both in the stimulated hemisphere and in the hemisphere controlled under stimulation. And in fact, as you can see, the red line indicates the tEPs evoked at the trough, and the blue line, the tEPs evoked at the no trough. The tEPs were actually greater when stimulation happened at the high excitability state. And this was true also up to 130 milliseconds post-pulse. So this means that the differentiation was pretty preserved in the unaffected hemisphere of stroke patients. And then if we are right, and the differentiation is really preserved within the unaffected hemisphere, then we should also see stronger post-pulse alpha power when stimulation happens at the trough. And this is exactly what we found. In the unaffected hemisphere stimulation, the post-pulse alpha power from immediately after the pulse increases all over the cortex much more compared to if the stimulation had happened at the low excitability state. But then let's go look into the affected hemisphere, in which we usually, without a phase-dependent approach, we see this increased response, right? And what we saw in the affected hemisphere is that yes, the differentiation is still there, but as you can see, the tEPs evoked at the no trough, so the blue line, are much more similar to the tEPs evoked at the trough. So this means, this suggested us, let's say, that the differentiation or the degree of differentiation between the excitability states in the affected hemisphere was less preserved. And therefore, when we went looking into the post-pulse alpha power, we wondered whether we could find this post-pulse alpha power modulation depending on the brain state at the moment of stimulation. And this is what we found. So in the affected hemisphere stimulation, up to 150 milliseconds, we do not see any post-pulse alpha power depending on the excitability at the moment of stimulation. So Just to wrap it up, previous works found already these greater responses in the affected hemisphere of severely affected stroke patients. So this is Sara, for example, we have Chairperson, but also other works. What we suggest with our results is that this increased response in the affected hemisphere is due to a lower degree of differentiation between the excitability states. So basically the affected hemisphere is more excitable also at what should be the low excitability state. And exactly what we're doing now is to pursue the investigation of whether the degree of differentiation between the excitability states in the affected hemisphere predicts the levels of impairment in the stroke patients. So these are just a couple of references if you're interested in the topic of inter- and intra-individual variability in stimulation studies. And I thank you very much for your attention.
Thank you, Arianna. Is there any question? If not, I think we'll keep them also later. And we can now move to the next speaker. Next speaker is Marta Bortoletto. Marta is the leader of the neurophysiology lab at the Pavia in Brescia. She's interested in the role of effective connectivity in cognitive functioning in healthy and pathological brains. The title of her work talk is Measuring Cortico-Cortical Effective Connectivity from TMS-evoked potentials with perspectives and challenges.
Thank you. Well, I think from the previous talks, we already know the relevant information about the TMS-evoked potentials. I'm also going to talk about TMS-evoked potentials. In this case, sorry, we are looking at TMS-evoked potentials as measures of effective connectivity. In several studies that are published, TMS-evoked potentials are interpreted as measures of cortical excitability. And that is indeed a type of interpretation that we can give. We stimulate, and then we measure how much the cortex responds. But if we look at more features of the TPs and its components, we might extract more information that is related to the connectivity of the cortex, and more specifically on the connectivity of the target area. And our studies that we run in my lab are at the moment aimed at validating TMS-evoked potential components as measures of direct effective connectivity. Why should we do that? At the moment, we have several different methodologies that we can use to measure effective connectivity, like fMRI or MEG or EEG. But to get information on the effective connectivity, which is the connectivity with the direction between areas, we need to use very complex models. So we thought that with TPs, we can actually have a different way to calculate effective connectivity, which is less based on computational methods. Okay, so in our labs, we have focused our studies mainly on the primary motor cortex. And our idea is that when we look at the components of the TPs, we are actually looking at activation that spread from the target area to connected areas. And by looking at the different latencies, we can distinguish different moments of the spread of activation in the cortex. If we want to look at the direct connection of between two areas, we should look at the very early components, the first one that are elicited by the stimulation. And by looking at these components, we might get information that we don't get from other techniques. What kind of information can we get? First, we have a measure of amplitude, which is basically how much the connected area responds to the stimulation. And then we can have measure of latency. So how fast the signal has traveled to a connection to reach a connected area. So this concept is similar to the way we currently use MEPs to study the connectivity of the corticospinal tract. You know, when we stimulate the primary motor cortex, we elicit MEPs in the muscle, and the amplitude of the MEP indicates how much the corticospinal tract has been activated, while the latency of the MEPs indicate how fast the signal has traveled through the tract. We can just do the same with the EPs, considering different areas. So we calculate the amplitude and the latency of the EPs to calculate the strength and the velocity of signal transmission. Specifically, we have, as I said, we focus on the primary motor cortex and the interhemispheric connectivity through the corpus callosum. This is because the function of the corpus callosum is very little understood. Even if it's a very big structure in the brain, we still don't know exactly what is its function and how it affects the hemispheric spatialization. And there is like a theory that has been proposed long ago that proposes that that the hemispheric dominance is actually related to the time that the signal takes to travel through the corpus callosum. So because the corpus callosum has a long time delay for signal transmission, that is the reason why we have a specialized hemisphere. But this theory has not been tested so far. Another thing is that, well, we have heard Arianna talk about variability in the subjects, and we do have inter-individual variability in our connections. So when we apply TMS to actually induce plasticity paradigms, there is variability also in the connection that at the moment we are not taking into account. So if we, like, for example, when we apply CC-pass paradigms in which we stimulated the two different hemispheres, we never take into consideration what is the time that the signal takes to travel between hemispheres. But if we can measure this latency, we will be able to implement personalized paradigms. Yes. I like, Luigi, that you say yes. Okay, so far there have been estimates of the signal transmission time between the hemisphere, between the two motor cortices from different methodologies. And the estimated time is between 10 and 20 milliseconds. And specifically, I want you to focus on the ipsilateral silent period because this is a measure that we use in our studies. In this case, the stimulation is applied on the primary motor cortex, and it is possible to see a reduction in the muscle contraction in the ipsilateral hand. This is because the activation of the stimulated area has traveled to the contralateral N1 and it induces inhibition. And we can see then the inhibition traveling to the corticospinal tract and affecting the signal in the muscle. So we have used this measure to validate the components of TPs that might reflect the transcallosal signal transmission. Okay, so in our study, basically we we recorded TMS-EEG from stimulating the primary motor cortex while the participants were contracting their hand so that we could measure the ipsilateral silent period, and we wanted to know which signal in the TP components might reflect a transcallosal signal transmission and the activation of the contralateral M1. And here you can see the TPs that we obtain. And specifically, we found that at 50 milliseconds, there is a component which is positive on the contralateral hemisphere. This component is reported in several papers already, but usually people tend to interpret it as a single dipole coming from the hemisphere ipsilateral to the stimulation. In our studies, we wanted to test an alternative hypothesis that this positive component is actually something separate and comes from the contralateral motor areas. And actually, we also found that some time ago, in 2008, there was a study with intracranial recordings in which they found a positive component around the same latency at 15 milliseconds in the contralateral hemisphere. So in this case, we tested if there is a relationship between the amplitude of the what we call the P15 component and the strength of the ipsilateral signaling period recorded in the muscle. And as you can see, the relationship is significant and is positive, which means that the higher the P15, the higher is the inhibition measure in the muscles. And we have replicated now this same effect in 3 different datasets, which are published or under review as a pre-registration or register reports, just to link to what we were saying this morning. Also, we also ran another study in which we always stimulated the M1, and we again recorded TPs, but in this case, we wanted to test if the P15 is a contralateral by looking at the functional connectivity recorded in the resting state before, in the baseline period before the stimulation. So in this case, we again measure the positive component, and we found that you can find here, and we found that this positive component is positively associated with the phase synchrony between the 2 hemispheres in the alpha phase. And this also links very well with what Arianna said before about the role of alpha phase in the connectivity between contralateral motor areas. Then in another study, we look at a different validation. We measured the microstructural integrity of the corpus callosum, and specifically the body of the corpus callosum, which is the part that connects the motor areas. And in this case, we found that there is a relationship between the integrity of the structure of the corpus callosum and the latency of the component at 50 milliseconds, which again supports the idea that this component is associated with the corpus callosum and the transfer through the hemispheres. So this series of studies can support that early TPs provide a measure of direct connectivity between the motor areas. And now we are investigating if we can use this information to individualize plasticity protocols. But this is the motivating part. We actually found also that it's not so easy to measure this component. We know if you use TMS-EEG, you know that this technique has several artifacts. The signal is covered by several artifacts, mainly in the early part of the epoch, where we actually measure this component. But also there are like parameters of the TMS that can change the features of this component. Specifically, we wanted to see how the P15 and our measure of interhemispheric connection can change depending on the current shape and current direction that we give to the M1. So we run in this study, we use both monophasic and biphasic stimulators with 3 different current direction. The PA current direction is the one that we know is more effective, is the most effective one to activate the M1, but also we know that other coil orientation can still induce MEPs and M1 activation. So in this case, I'll just go directly to the result. As you can see, when we use a biphasic stimulator, we always obtain the P15, but and the amplitude is not different between conditions. But in this case, if we want to measure and estimate the signal transmission time between hemispheres, this is the latency of the component is actually affected by the coil orientation, and it is delayed for the AP current direction. Yes, which is. While when we use monophasic stimulator, there is actually a huge alteration of this component because here you can see that for LM and for PA condition, we basically don't have the P15 component, while for the AP condition, the component is extremely high, and again, the latency is delayed, which means that if we, for M1, the variability, the anatomical variability is not that much between subjects, but if we want to use the same approach for different areas where there is more variability and we don't know exactly how to rotate the coil, it might be harder to get this information across participants. So to conclude, we can say that early TMS-evoked potentials can provide proxies of direct effective connection with specific features that are not possible to measure with different techniques, and they allow to estimate the conduction delay. But on the other side, they are heavily affected by stimulation parameters, and we do need to understand how we can obtain reliable measures across participants. And well, I haven't talked so much about the artifacts, but of course we need to have very careful cleaning of the data because we have big artifacts in that part of the epoch. So to conclude, I just want to thank all my collaborators in the lab who contributed to these studies.
Is there any question? Well, not for now, for data. And now we are moving to the next 2 talks, which are online. Right? Next speaker is Doris Pischedda. Her research interest, well, Doris, she's an assistant professor at the University of Pavia. Her research interest is in how to use brain parameters from fMRI to build personalized virtual brains. But she's also interested in gender bias in neuroscience. And today she's here with us to talk about the women in neuroscience.
Thanks for the introduction. Can you hear me?
Yeah.
Okay. And can you see my screen?
Yes.
Perfect. Okay. So good afternoon, everyone. Today, as was said, I'm talking about the Women in Neuroscience Repository, which is our initiative. But let's start from the basics. So why did we start this initiative? So as is pretty clear to everyone, we know that there is a gender bias in academia and also in neuroscience. And on one side, you can see that there is a gender pay gap. So basically, women are paid much less than their colleagues men. And also there is a higher career reaching rate for female postdoc, which means that the gap increases with the seniority of the academic position. So at the lower level, so undergraduates and PhD students, you have more or less a balanced number of male and female. But as long as you go up in the scale, at the senior position and professor in particular, you have much less percentage of female on that positions. And there is a lot of research done on this topic, and it's been shown that, for example, there is a lower acceptance of paper that has a female last author. There is also lower recognition of the contribution of women in papers. They are also less prone to get fundings, and there is a lower range of invitation for them at conferences and workshops. And also there is lower chance to be hired for a tenured position. That's why there is this increasing gap in the higher positions. And this somehow is also related to the fact that women are underrepresented in the peer review process and also in deciding bodies, because there is this bias, which is like homophily. So both women and men prefer their same sex. If you have more male in these kind of committees, then men will get more prizes, more acceptance, and more fundings. Okay, so just some facts about this bias. So in 2019, we published this paper, which is called Gender Bias in Neuroscience, Facts, Consequences, and Solutions. And in this paper, we look at the representation of women in different aspects of the academic life. And using data that we found online, we could see that, for example, while it's kind of the same amount of women and men that are present as first author in accepted paper, if you look at last author, you have many more men. And the same holds for prizes. So men get more prizes than women, and they're also more often invited as a speaker or at seminar or conferences. So these are the facts. And of course, this has consequences on research. First of all, if a group is not very much diverse, the group is less innovative, less flexible, and it has also more problems in finding problems, in solving the problems. So the more diverse a group is, the easier it is to find a solution, because people can see the problem from different angles and they confront each other, and it's easier to find a solution. And also brings to the underrepresentation of women as a target population in biomedical research, because most of the time the studies are done using men as a population, which means that whatever treatment is developed or a prevention program are developed, it's much more difficult. They are effective on women if they were not like the part of the sample from which the techniques were developed. And also, if we mix an equal proportion of male and female, then the results of the study will be biased. So it will be good to have only female or only male groups, or of course, the best would be to have a balanced number of the two genders in the research to avoid this kind of underrepresentation. And we've also seen that the bias holds also for AI. So usually AI models rely on data that are collected from clinical population, which consists most of male population and white male population. And so if we use this kind of model and the model have a bias in the data they use, also the result would be biased. So this may not be useful indeed for the real clinical settings. So in the paper, we also propose some possible solutions at different levels. So we have at the institutional level, the organization level, and the individual level. Here you see a list. I will just focus and highlight some of them. So for example, at the institution level, institution can collect data and share it and try to assess how well their policies worked in reducing the biases. At the organization level, you could search for candidates on list of repositories where you find women, or you can also provide childcare and other family-friendly measures so that women are more willing to participate or are actually more able to participate in this kind of events. And at an individual level, female can sign up on repositories, or people can encourage their colleagues to do so. So here you find some resources and databases on bias and diversity. So the first one, Bias Watch Neuro, basically is a website where you find, just keeps track of how many women and female are invited as a speaker. And still, if you look at it, you will find some events where you only have male invited as a speaker. And here you find other sources, which are more lists that you can consult to find, for example, suitable female for the speaker, or if you want to look for candidates. And they also find our repository here. And there is also some initiative that looks at policy debates on gender balance. So now I will talk to you about our initiative, which is the Women in Neuroscience repository. So this is our website, and here you find the board members of the website. So basically everything happened and was started by Jessica, which was at one of the conferences in neuroscience, and she noticed that there was no single woman invited as a speaker. And so she started wondering, how can we help that? And then she said, okay, maybe if there is a repository in which people can look to find people they can invite to the conference as a speaker, it would be easier for the organizer of the committee to find more women. Okay. And so these are just some general features of the repository. So we have almost 2,000 profiles. You can see here like the distribution by seniority, and also the geographical distribution of our members. You can also sign up and be part of it and create your profile on it. You can look for profiles using specific keywords, for example, themes or techniques as a filter. And then you can submit a recommendation for someone you know, for example. Then another feature that we have is the profiles. So here you can search for someone you're interested in.
Sorry, Doris, could you give us a moment, please?
Yeah, sure.
Okay. Yeah, yeah. So you can go on, sorry for that.
Okay, no problem. So I was saying you can search for someone here. You can also restrict the search for underrepresented countries, for example, or just senior position. And then you will see the results of the search. And if you go on view profile, you will see the profile of the person with some links that may be useful for you. And there is also a feature which is called the recommendation feature. And then people can write recommendation on the person, so that when people are looking for suitable candidates for as a speakers, they already see some information provided by other people, and they can get an idea of how the person is and what kind of things she does. We have recently introduced the account in the website. So everyone can create an account on the website. You can do it on form or just using your socials. And this gives people control of the data, so they can create the account and they can update their profiles. This is for women, so you can have profiles of women neuroscientists, but everyone can have an account, and they can write a recommendation to them. So if they know someone and they want to recommend them, they can just do it. There were some existing profile in the past, and this can be claimed. So if someone signed up for the repository in the past and they want to claim the profile, they can simply create an account and then follow the instruction to claim the public profile. And of course, new people can sign up and create their own one. Another improvement that was added to the website is the publication list. So here you can find a list of publications related to the topics we're interested in, and you can have different type of it, and you can look for them. And this is just a list that is on in this moment. And what we are also working on for the website is we want to add an event page, and there will be linked to the notes we took from the past event we organized, but also links to sign up for the new events, and a new page where we advertise jobs and grants that are targeted for women specifically. And also we want to implement a new recommendation system, because at the moment it's something like that. So you have something to choose, but most of the stuff will be written by you fully. And what we want to have instead is a semi-structured questionnaire so that people can simply choose options and maybe add very little tests, so that maybe people are more prone to leave a recommendation than we'll have more recommendation on the website. So if you want to contribute to the development of the website, we have our GitHub repository, so people can go there and look for the validated issues we have, and they can follow these guidelines if they want to contribute to it. So we have different open fronts for our projects. So beyond the website development, we are also interested in research on gender bias. And we also try to promote these kind of topics and communicate about it, both on the socials and conferences. And then we also open for collaboration with other groups. And of course, you can join us if you like. And as for the long-term goals, we would like to collaborate and create a network to share contact on EDI, and also to partner with other EDI groups. And here you see just some of them that we already contacted. So before I mention the events, so we usually organize these informal panels, and then we share recommendation on them. We just talk to each other. And here you see the teams of the events we organized last year. And the next one will be next month. So there is an event on intersectionality in neuroscience. Here we see the list of the panelists. And of course, you can sign up if you want, and you can just go there, ask your question, share your tips or whatever. Okay, so to conclude, for women neuroscientists, you can create an account on the repository. The organizing committee can use the repository to find profiles that are suitable as speakers for their conferences. Everyone can submit recommendation on the website, and you can also spread the word. So on the website, there is a slide that you can download, and you can put it at the end of your presentation if you want to support the initiative. And most important, we are looking for volunteers, because we are all volunteers in this project. So if you would like to help in any of the topics and the aspects that I showed you before, you can either send us an email, or you can just fill in this form, and then we will get back to you. Thank you for your attention, and I'm happy to answer your question. And also the slide will be available on Figshare if you're interested very soon.
Thank you, Doris. Is there any question? If not, I had the curiosity more than a question. I was wondering if, well, I don't know how long the repository has been active, so I'm not sure whether you have enough time to collect this information, but I was wondering if you have any way to kind of monitor whether organizers of conferences are actually looking at the repository to find speakers.
Yeah, so this is part of the research part that we are doing. So we just use some simple Google Analytics and just talking to people that are organizing the conference. We know that some of them, they use it actually. So this is on since 2018. So it's already a few years that it's there. And we know from some people that they actually use it when they were organizing the conferences, but we don't know exactly because we just have information about how many people go on the website, but we don't know if they go there just to sign up, to look for it, or just to really use it for their purposes, for the conferences. But this is like one of the projects we want to start.
Thank you.
Okay.
Yep. Yeah. Carlo?
I'm coming there because otherwise we won't be. Thank you, Doris. This is Carlo. I was wondering, since I've used it before, there's also a repository about non-invasive stimulation. Since we are discussing about non-invasive, I was wondering if you can sort of create links or eventually ask to that people organizing the repository to merge information between the two. That would, sorry, that would be helpful, I guess. So it will also help to potentiate the diffusion of information.
Yes, I totally agree with that. We tried with other groups to share and to try to find a solution to have a like common repository so that people don't have to look too many websites. We couldn't do it yet, but we have some collaboration with this, and I would be happy to contact these people as well and then see if they want to do it. But most of the time we have on our website links to the other repositories so that people, if they look for very specific type of speakers, they can do that. Okay. But of course, having everything in a single website would be much easier for the organizers.
Yeah. Thank you.
Thank you, Doris. I guess we move to the next speaker now, and the last speaker. Last speaker is Jovana Bjekic. Sorry if I spell it wrongly. Jovana is an associate research professor at the University of Belgrade, and she's the co-lead of the TwinNIBS project. Her research interest is in the application of personalized NIBS to target memory and other cognitive function, but today she will discuss with us the gender dimension of the TwinNIBS project.
Hi, thank you. Can you hear me?
Yes.
Yeah, great. And I managed to share my screen. Yeah, right.
Yeah, we can see it.
Yeah, thank you. Thank you for the introduction, and the name was perfect, so thank you. Yeah, as you said, I usually talk more about brain stimulation and how it affects cognitive functions, but today I will be talking about the gender dimension in non-invasive brain stimulation. So I'll essentially take a step back and discuss how did we incorporate the gender dimension into the TwinNIBS project that organizes this workshop. So I will essentially talk about two streams that we actually saw today. One is the research. So how do we integrate sex and gender aspects into our scientific practices? And then I will talk about the researchers. So there I will piggyback much on what Doris just said. So starting with the research part, just let me give you a brief background. So the main issue here is that most of the things that we know about diagnosis, treatment, prevention of disease originates from the studies mostly done on male cells, mice, and human men. So a recent review of basic biomedical studies showed that 80% of publication included only males, either human or animals. This essentially stems from the two misconceptions. The first one being that female animals or humans simply increase experimental variability due to hormonal fluctuations. What we are now painfully aware of is that the one-size-fits-all approach does not work, even regardless of the gender. So that we might need to go down to individualized approach rather than using a single model to model behavioral and brain in general. And the second one is the historical belief that there are essentially no major differences between sexes apart from reproductive functions. This actually translates directly into our research decisions. And if you look at the works that analyzed work published in top 6 neuroscience journals just in 2017, we see that the research done on rats and mice are either 50% of them are done on male model only, or 1/3 was done on male model only. And this is what we usually regard to as bias, meaning that what we know about the basic mechanisms, we actually know about the basic mechanisms in male model. Moving on to the human studies, we do not have that type of discrepancy, even though we have some male-only studies, but we have more of what's called omission, meaning that both sexes are included in the study. However, sex or gender is actually not considered at all at the analysis level or data reporting. So it's essentially a neglected variable. How does that translate to non-invasive brain stimulation research? So as in other neuroscience field, in NIBS, we usually involve both male and female participants. However, we do not take into account participant sex when doing data reporting on or the analysis. Does that have the real effect on our outcome? Actually does. So looking at the example for TMS treatment for pharmacologically resistant depression, there was an analysis of patient outcome of more than 5,000 individual data, which found that when controlling for various protocols and symptom parameters, females were 1.34 times more likely to respond and 1.30 times more likely to achieve remission from major depression relative to males. Also, another meta-analysis performed over the span of, so the research spanning the 16 years and 54 sham control studies found a positive linear relationship between the percentage of females enrolled in the clinical trial and the overall reduction of depression severity. Therefore, the conclusion here is that women tend to respond better to TMS depression treatment. There have been 3 hypotheses lately put forward why this might be the case. First one is bone structure differences in males and females that influenced the actual electric field delivered to the scalp. In essence, we are basing the dosage of TMS based on the motor threshold. And we have differences between males and females in the distance between the scalp and the brain in the prefrontal cortex, which is much larger than the one at the motor cortex. Therefore, effectively, on average, women tend to actually receive higher dosage of TMS than men, even when that dosage being mapped to motor threshold. The second hypothesis is that there are differences in gray and white matter, so in the neuroanatomy of the brain. And I will not talk in detail about this because from what is available there in literature, it seems that this is less a sex issue, but more individual differences issue, which then reflect on the group level on the sex differences. And finally, the third hypothesis, which is of course compatible and complementary to the previous two, is the hormonal differences, which are definitely sex relevant. In essence, there are studies that found that at times of high estradiol and low progesterone, we have increased cortical excitability. This is the variability that drew researchers away from a female model earlier. But now what we know, for example, and where this can be used is when we have the accelerated TMS protocols that Armita was talking about. Actually, there was a study showing that those protocols might be more effective if delivered at a certain levels of hormonal differences. Therefore, we can use this information to actually increase the effectiveness of our treatments. So based on this, I highlighted some of the key recommendations because there are some early stage researchers today here with us. So some practical recommendations for conducting sex and gender sensitive research, which we are actually implementing in our TwinNIBS project and in general in our approach. First is enrollment of both male and female participants to the study. But also keeping in mind that we need to have equal female-male allocation to the experimental conditions, trial arms, so that we can avoid biases that stem from gender in terms of the results. Also taking the individualized approach to dosing, meaning intensity, location for NIBS delivery and montage, and also being mindful about potential gender biases at the outcome measures or gender differences in symptoms. To simplify this, Dunja was showing the tasks, and you saw female, male faces and so on. We know that there is a gender bias of better remembering the same sex. Therefore, we need to be very careful how do we use the stimuli and balance that across conditions as well. And I think here is an important part, and that is there is a strong push for analyzing sex and gender differences whenever we collect data. We all know that we are working with small samples. Therefore, we are often not sufficiently powered to actually do these type of analysis. So I would like to advise against doing it to avoid type 2 error. However, it would be great to go back to the open science approach that we discussed this morning, that we actually need to share the data so that we can collide them and have prospective multi-data set analysis which are sufficiently powered. That would be the research aspect. Now I'm moving on to the researchers. So Doris said most of it, so I will just be repeating and rephrasing. So there is the main issue here is the glass ceiling effect, which boils down to the fact that the proportion of students in STEM at the undergraduate level are similar between men and women. However, the gap between male and female representation increases with the seniority of the position. There are many evidence corroborating this. I just took out two here. So in 2003, we had 50% of female PhD students, and then moving up 25% of tenure track faculty and 22% of full professors. 15 years later, essentially nothing changed. 53% of PhD students, 30% of tenure track faculty, and 30% of full tenure professor. This is focusing on neurology and neuroscience. I will highlight again just the graph that Doris showed from their paper, which I think outlines very illustratively what's happening. So we have equal distribution between men and women when we look at the first authorship on the paper. We have more men being last author than women, so senior author. However, where I think the biggest gap actually happens is that the gap in conference speakers actually supersedes the senior authorship issue. And as you can see, there is a significant amount of all male conferences out there on the graph that were found. For those of you that are new to non-invasive brain stimulation community, I will just walk you through how this was happening in the brain stimulation conference. And I just want to say I'm telling you this story because the take-home message is that things do get better. So in 2015, there was a first International Brain Stimulation Conference organized by Brain Stimulation Journal. And there were 2 out of 39 invited speakers who were female. Next conference was in Barcelona, 2017. And 100% of invited speakers were men. This sparked a response. Kate Hoy wrote a letter to editor to the Brain Stimulation Journal titled Gender Imbalance at Brain Stimulation Conferences. We have a problem that's everyone a problem. And this one got a lot of reactions. So one of the responses was that Bias Watch Neuroscience rated the conference 0, meaning that the percentage of female speakers was more than 2 standard deviations below the estimated percent of women in the field, which was 34 at the time. Then Mark George and Sakim, so the editor-in-chief of Brain Stimulation, the organizer of the conference, wrote a response to Roy, essentially saying that the highest priority was and will continue to be a scientific merit. And the diversity is important, but it's not only gender, it is brain stimulation techniques, disciplines, geographical area, clinical research, and so on. But the main defense was that it's based on scientific merit, not on gender bias. However, a careful analysis and Hinder and colleagues wrote a response to the response. And actually, they audited the research articles published in Brain Stimulation Journal between 2014 and 2016, so essentially preceding the conferences, and shown that 30% of first and senior authors were female. And when they selected the most highly cited paper from each year, actually 35% of those papers were female first or last authors. Therefore, the scientific merit did not really reflect in who was invited to speak. Meanwhile, the second European Conference in Brain Stimulation in Psychiatry actually responded by having 40% of female speakers and addressing the importance of gender balance at the opening session of the conference. And actually, they wrote a really nice editorial to the brain stimulation, explaining and highlighting how they address the gender issues in their organization of the conferences. So essentially giving us how did they achieve this. As I promised, things do get better. So Third International Brain Stimulation Conference 2019, 4 out of 11 plenary were female. 2021, we had the Charleston conference. So this is like a post-COVID, half-COVID conference. We had 5 out of 12 plenary speakers who are female. And just this year in Lisbon, we came to 7 out of 13 plenary speakers who were female. Essentially, we came to the 50-50 at the plenary level. So this was just the trajectory. Showing that it can change, but there are collective actions to be taken. Therefore, I will jump to the recommendations. So, as Doris mentioned, there is a repository which is for whole neuroscience, but Carlo just mentioned the one that is specific for brain stimulation. So it is Women in Brain Stimulation database, which was actually developed at the time and by the group when the Kate Hoy letter happened. So she actually started this initiative. And the problem there is I'm not seeing how effectively this database is used. Therefore, it exists, but there is a road ahead on what we do with it. So sign up. And the second actually recommendation is what we tend to call speak and attend, meaning that women tend to opt for poster presentation more often than men. So whenever have a chance, take it and speak. And also it is good to show support and attend female plenary lectures and so on, because the attendance is what drives the interest essentially. If you want to see which conferences or journals have what percentages and how it works, so it's a Brainwatch Neuro, and essentially none of us individually can change like the landscape tomorrow, but it is very important to be mindful about gender bias in the field and take a small steps to contribute to the diversity and equal opportunities. Speaking of small steps, this is my disclaimer. So the TwinNIBS project, which gathers all of us here today, has 1 female out of 9 at a leading position. So we also have 40% of female researchers across the whole project. Therefore, we don't have it figured out. I'm just putting it out there. But however, we launched the Bootcamp Research Project, which is actually a mentorship program for developing internationally collaborative project in non-invasive brain stimulation, where we had 3 out of 5 mentors, so leads, who are female. One of them is with us today, so Armita is leading her group. And we also have 52% of mentees who are female. Therefore, we will probably see the products of this project being far less biased in terms of gender than what's currently happening in the field. Thank you. Yes, this was a quick overview of how we do it, and thanks.
Thank you. I guess now we finished with the talk. So what I would like to do is just to make sure that all the speakers are here, the one in person and also the one online. I will just briefly summarize my take on today's talk. So the first thing is that I guess we've seen the importance of incorporating somehow individual variability in both NIBS protocol and also data analysis. And this variability can be at several levels, from the anatomical level to the functional level and behavioral level. So one question that I have in relation to this is, well, it's for all the speakers actually, how far do you think we are from connecting this variability, which, as I said, it's on many different levels, to actual higher cognitive performance? And if you think, how do you think we can improve cooperation and collaboration with different laboratories to try to address all these different aspects of variabilities? Is there anyone who would like to? Arianna? Yes, please. Yes. Yeah, maybe it's easier. So yeah, it's easier so the microphone is here. There is one chair here. Yeah. So you can take this one. So I'll start. Yeah. So the question was how far we are from integrating these concepts and this control, let's say, in the study of higher cognitive functions. I mean, I speak from the brain state dependent view. I guess cognitive functions are very much linked to networks, and controlling for networks means that we need technical, practical ways to target networks, which is not very easy, as merely you can, like, it's very hard to do, for example, connectivity trial by trial. So I think
that is like if we manage to develop techniques that can target and individualize in real-time networks, then in that sense, I think we would be much closer to understand what cognitive functions are. And what is the relationship between structure and function in that sense, and also how to decrease variability in cognitive function in studies investigating cognitive functions in general. But I think that without the technical aspects, we cannot really answer the questions or fill the gap, let's say, in the knowledge.
Yeah, so I would add that, well, my feeling is that from the technical point of view, researchers have done like huge step forwards. Like even from the talks today, we heard so many exciting and new way to control several different aspects of variability, which is great. So I think we are close from the technical point of view. But on the other side, we need to make an effort to have like very big collaborative projects where we can collect thousands of individuals. In fact, there was a paper, which I'm sorry, I don't remember the first authors, but they basically showed that if we want to control for individual variability in testing the relationship between brain and behavior, especially for high function, we need 10,000 participants. So I think the real effort now is to, work together, basically, in big collaborations.
I guess this goes in the same direction of what Giovanna was saying, that data should be made freely available or in open data sets. Do you want to comment on that, Giovanna?
Yeah, thank you. Thank you. So my view on the question you posed is that we essentially have the individual differences at so many levels, from the structural one, which Armita was talking about, then to the functional one, which also Armita and Dunja were talking about, then coming down to the state dependency, which was also addressed by Arianna. The part which we did not actually tackle a lot today is at the outcome level, what happens. So what measurements are we using actually to see the change, meaning the reliability of the tasks we are using, the representativeness, and so on and so on. Therefore, I think with all those sources of variability that can be there, tackling them in a single study is essentially impossible. Therefore, what we can do is use individual studies to assess very specific hypotheses and factors that may contribute or does not contribute. But what we need is a collaborative effort and essentially data sharing in order to effectively estimate the effect size of each of those variables. So some variables actually might not have an effect, even though we assume they do, or that effect might be too little. So I think that's very difficult, what you're, for example, trying to do in multi-site TMS studies. That is a huge collective effort, which is applause all the way around. But there are like even easier stuff that can be done, which is, for example, sharing the data sets on gender and see how much sex differences are there, or age, or tiredness, or all those variables that we are taking. All of us are registering it, but we're never analyzing it per se.
Thank you. I see Doris, you want to add something? Sorry, we can't hear you.
Yeah, I totally agree with what was says. And I think it's very, very, very important point that you are talking about. And so now I'm part of a very big project, which tries to look at models that we can use in health and disease. And we also are focusing a lot on individual differences. So basically, we try to extract parameters from the subject measure, MRI measures, and to simulate the brains. So we use this virtual brain to try to create a double of the brain that is specific for the subject, so that it's easier and more effective to use for prediction, for example. So I think this is very, very important. And the nice thing is that now people know it, and you see many more bigger initiatives that tries to network together and solve this kind of problem. So I just wanted to point it out, that is very, very useful.
Thank you. Is there anyone here or online who would like to add something?
Yeah, can you hear me now?
Yeah, yes.
Yeah, I just want to also say that I do agree with all the comments that has been shared so far. Especially, I think that we can divide it in two parts. First, before conducting the experiment, just considering that how we can, what are the strategy that we can take and take the actions to minimize the inter-individual variability or intra-individual variability. So it can be I have seen the checklist that we can consider before the experiment. For example, if you want to go more scientifically, it's about the sleep quality, the hormones level, the gender, the how many subjects you want to cover. And then the second part can be what Giovanna and Doris suggested, it's the transparency, to just share the data, have the higher number of the subjects when we are going to compare. And this way, I think that we can get closer to addressing this problem.
Thank you. Well, the next point that we touched was related to gender-informed scientific practices. This is kind of often overlooked, and we saw that it's an important topic. Jovana, you presented some ways you're incorporating, or Twin NIBS is incorporating gender-informed practices in the project. I was wondering, in your experience, what do you think is the aspect that researchers are more reluctant to adopt, and why, if you can speculate on why?
Yeah, thanks for this question. I actually do have an opinion on this. So I will just take a minute, because I think sometimes when I started working, whenever you saw a tutorial for EEG or for brain stimulation, it was done on a very round, nice, almost short hair men, because it's easier to see electrodes, it's easier to put gel there, it's just easier to manipulate with. And what we saw actually in when you talk to people, what you see is that they don't know how to handle difficult subjects, meaning Black people, women with a lot of hair, and so on. So I think the reluctance to use these difficult subjects is not because somebody doesn't want them, it's just more difficult. And I don't know if you watch those tutorials, but have you ever seen men who are treating women's hair with trying to put the electrode? That's almost like a funny video. Therefore, I think a way forward there is actually what Black in Neuro actually did it. It did a tutorial on how to put an EEG cap on black hair, which is very useful for people who do not have an experience with it. We also did a tutorial on how you place the electrodes, and we usually use the female model to show it, because there are actually additional steps. So therefore, I think that tackling these issues is essentially just teaching people how to work with those difficult subjects, rather than just highlighting the problem and asking, presuming that there is a bias there. I think it's just a level of competence that we can, increase, and then it will work just fine.
Thank you. It's very interesting. I don't see if there are questions. I don't think there are questions from people online. I don't see anyone.
So if someone is raising the
yeah, it should come up first. Yeah.
We're going to see it in the
So I don't know if there is anyone who would like to comment. Carlo?
Personal comment. It's much more difficult to get a good impedance in the man. Actually, much easier with the girl.
I
Yes.
agree.
When I was trying to put an EEG cap in a girl with the straight dreadlocks, I didn't realize that it took me 2 hours, but no, but it's much easier, especially when you try to put a cap in a bald guy.
Yeah, that's a nightmare. That's a nightmare. But that's true. It's I agree.
Because they carry the marks for days after that. They have the marks on their head if they are bored for days, maybe.
Yeah, they don't think.
Yeah, it's well, I guess if there are no other questions or comment, we can conclude the symposium. I would like to thank again all the speakers and the attendees for being here today, and the organizer, of course, and the twin NIBS. This is really a tongue twisters, this name. The project. Thank you very much.
Thank you for hosting it.
Thank you.
Thank you.
Thanks.
It's better than that's my goal. Thank you.
Actually, it was good also. Yeah, just